Chapter 2, The Chemistry of Living Things, Screencast 5. Now, this is the last screencast for Chapter 2, and it's going to be focusing on two things, proteins and nucleic acids. Now, I don't like playing favorites, but if I had to pick the most important of all biological molecules, I'm going to go with protein. My training is as a physiologist, and really a lot of it is just proteins, the study of proteins and protein activity, all of those things. They are also the most complex, probably, of the biological molecules aside from DNA. Now, you'll hear the term polymer. We saw this previously when we looked at polysaccharides, so long chains of glucose molecules. Here, the building block of proteins are amino acids. There are 20 different amino acids, and they have an amino end. That's where the nitrogen is. So amino, nitrogen, carboxyl, that's the acid end, and an R group. So all amino acids have these three things in common, and what makes the actual amino acid different is the R group. Now, amino acids are joined together by peptide bonds, and they're produced by dehydration synthesis reactions, which we talked about previously. Now, take a look at the number 20. Okay? As I said before, amino acids are the building blocks for proteins. Imagine that alphabet comparable to the English alphabet, 20 letters, and think about all of the arrangements and combinations that can be made. So that really talks about the diversity of proteins. Here we basically see individual amino acids linked together by peptide bonds and forming a polypeptide. Now, a peptide bond forms between the carboxyl end, that's the acid end, of one amino acid, and the amino end, where the nitrogen is, of the next amino acids. I don't want you to get caught up in the differentiation between polypeptide versus protein. Typically, I will refer to proteins as being relatively large molecules. The other thing that we'll talk about a little bit later is where do you expect to find proteins within the body? They're big, they're in a container to think of, okay, or a closed system. So proteins are found inside of the cell and also within our plasma. So in containers, so to speak. Now, one of the things about proteins that's really important is the relationship between structure and function. Proteins have a relatively complex structure, and all proteins have a minimum 3 general structures, and some complicated proteins or complex proteins actually have a 4th level. Now, the primary structure is nothing more than the amino acid sequence. And once again, that is linked amino acids through peptide bonds. Now we have a secondary structure. And there's 2 general shapes here, generally, that we will see for this secondary level of structure in proteins, an alpha helix or the beta pleated sheets. Now, the stabilization here is by hydrogen bonds. And remember, we introduced these earlier. Hydrogen bonds aren't strong, and we want to think of them as an attractive force. So that says something about the ease in which we can probably break these bonds, and at the same time, that protein is no longer going to work the way it should. The relationship between structure and function in proteins is critical. The 3rd level, or tertiary structure, is actually the 3-dimensional shape. This is also stabilized by hydrogen bonds. Now, what this is going to do is create polar and nonpolar areas within the molecule. Now, the 4th level of structure, known as quaternary, this is only found in those proteins that are made up of 2 or more polypeptide chains. Hemoglobin is the best example out there of a protein that has this 4th level of structure. Now, one of the things that we have to do within our body is keep a couple of things within narrow limits. Temperature and pH are 2 that we regulate very tightly. Now, think about what's responsible for holding a protein's structure. Hydrogen bonds. Do you think hydrogen bonds are going to be able to tolerate high temperature or low pHs or high pHs? And the answer to that is no. And since proteins are critical for function, our body regulates the environment that optimizes protein function. When a protein is disrupted either by temperature or changes in pH, basically that protein is now called denatured. And a denatured protein loses its biological function because its shape is no longer what it's supposed to be. Enzymes are a type of protein, and enzymes are very, very important because they're necessary for reactions to take place within the body. In chemistry, to get a lot of chemical reactions going in the lab, you use heat. We can't do that to people. And so there has to be something that's going to allow a chemical reaction to take place, and that's what enzymes do. So here's the enzyme, and we've got reactants. Now, this model is known as the induced fit model. Now, notice it's not a perfect fit between the reactants, and also notice what happens as the reactants interact with the protein. The enzyme basically changes its shape or conformation and allows the reaction to take place, and the product is released. So enzymes are a very important class of proteins that allow chemical reactions to take place within the human body. Now, if we look at the functional shape, this is something that I've talked about previously. Okay, so temperature, pH, the concentration of some ions, and there are some things will actually act as inhibitors, which will have impact on the shape and function of a particular enzyme here. Now, the next and last of the biological molecules are nucleic acids, and the role of nucleic acids is to store genetic information. Nucleic acids are long chains that contain subunits known as nucleotides. Now, if we look at the different types of nucleic acids, the two most common are going to be DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. DNA contains the instructions for producing RNA, which basically is going to be responsible for producing proteins within the body. A segment of DNA is known as a gene, and a gene is a segment of DNA that codes for a particular piece of RNA that will make a protein. So RNA is actually going to be the instructions, and RNA will move out into the cytoplasm and provide the protein factories the directions necessary to produce particular proteins. Now, as I noted before, proteins are probably the most important of all biological molecules because of the role they play within the body. So here we can kind of see the scheme. DNA produces RNA, which eventually produces proteins. It's a little simplistic, but it's just an overview of the process. Now, nucleotides are the building blocks of nucleic acids. And this is something you definitely need to know as far as what makes up a nucleotide or what do they contain. They have one 5-carbon sugar, and we talked about these previously when we did carbohydrates. So this is where we see them play a significant role. We look at deoxyribose, that's going to be the 5-carbon sugar in DNA. And we have ribose, which will be the 5-carbon sugar in RNA. They also, in addition to that 5-carbon sugar, will have a nitrogenous base and a phosphate group. So these are the things that are required to make up a nucleotide. So that's something I want you to be comfortable with. Now, the nitrogenous bases, I want to kind of point them out here. So I have adenine, thymine, cytosine, and guanine. Now, what's going to happen in DNA is 2 strands, and we're going to see something known as complementary base pairing. So the cytosine on one strand will complement the guanine on the second strand. We'll also see this complementary base pairing between adenine and thymine. So I want you to get that relationship. So adenine will base pair with thymine, cytosine will base pair with guanine. Now here we can basically see the historical classic structure of DNA as the double helix. It's double-stranded, and if we look at the nucleotides, they're going to be deoxyribose. We already indicated what those nitrogenous bases were: adenine, guanine, cytosine, and thymine. And I also kind of talked about the base pairing, and we can see that happening here. Adenine will base pair to thymine, guanine will base pair with cytosine. Now, RNA is a single-strand nucleic acid. So here, the sugar is ribose, and notice something's missing. There's no thymine here. So here, we have uracil instead of thymine. So when we're looking at RNA, looking to form a complement, adenine will complement uracil. So uracil is replacing thymine. So that's really important, and I want to make sure that you keep that in mind. Now, structure and function of ATP. ATP is a nucleotide. It's the cellular energy currency exchange. And the nucleotide here, adenosine triphosphate, and its bonds between phosphate groups contain a lot of energy. So breaking the bond in ATP is going to release energy. So here we have ATP, and then it's going to be hydrolyzed, and I'm going to have ADP, an inorganic phosphate, and energy is released. So that also is something really important that I want you to keep in mind. ATP we want to think of as that cellular energy currency. And that's what cells actually use. They don't use glucose. They don't use triglycerides. They're going to generate or make ATP from those other energy yielding molecules. Now here we can actually see the structure of ATP. So here's the three phosphate groups of the triphosphate, ribose, and adenosine. So that, once again, makes up, that's a nucleotide. All right. And what we're going to see here is basically how ATP is going to be hydrolyzed. So water is going to cleave one of the phosphate groups. Energy will be released. And then we have the synthesis side of things where a phosphate group is going to be added. So here's the dehydration synthesis side of things. Water will be given off as a product. And here we are back to ATP. So what we see occurring within the body is a constant cycle. Producing ATP, breaking it down, all necessary to maintain the energy required by our cells for normal function.